Hello there, it's Mrs. Scoble. Today we're going to be learning about how scientists test for mutations. Our lesson today has three objectives. The first is to identify the steps that are used to make a DNA fingerprint, and we'll see what that is, to interpret the aforementioned DNA fingerprint, and to identify chromosomal mutations, which we've already learned, by looking at a karyotype. Today we do need to start off with a very quick review, just reminding you, we get mutations when there are mistakes that are made in DNA replication, or we can also get them in transcription, and we also get them from environmental factors such as the sun, which again were called mutagens. We had overall several types of genetic mutations that could occur. First, we had gene mutations, which were small changes in one or just a few nucleotides, and those were point mutations or frame shift mutations. Recall that point mutations, we had three subtypes depending on what happens with the amino acids, and frame shift, you could either delete a nucleotide or insert one. Today we'll be thinking more about those chromosomal mutations as well, and actually we need to add one there, so remember those were translocations, inversions, deletions, duplications, and also non-disjunctions. Today, though, the focus of our lesson is to really think about how people actually test to figure out if there is a genetic mutation. And there are two main ways we're going to focus on. The first is DNA fingerprinting, and this helps see point and frame shift mutations in better detail. And then the second is karyotyping, which we're going to use for larger chromosomal mutations, like those deletions. And this will also let us see if a non-disjunction has occurred. So the first thing here, DNA fingerprinting. This is a process that scientists and also forensic scientists use to identify specific patterns within a DNA. So I've said here it's used in forensics, but this is an application that can be used for mutations as well. So similar to on your hands, you have fingerprints that are unique to you. Everyone's DNA is unique as well. As a result, in crime scene applications, in all cases, they'll have a marker that lays out specific sizes of those fragments in the DNA, and then they can collect a sample from the crime scene and compare that, to individuals that are suspects. This can be done as well for mutations, again, taking a known mutation and looking at it to see if someone has that mutation. DNA fingerprinting, in order to get that fingerprint that we just saw, kind of goes through four main steps. And the first is to actually collect the DNA from the person. And this is usually done either through a mouth swab with a Q-tip or a small blood sample. And then there are going to be more copies that are made using another process called PCR, which we'll talk about in just a minute. Then the DNA is cut into fragments. The DNA is very long, and it's cut into smaller base pair fragments. And these are going to be done using specific enzymes called restriction enzymes, and we'll go into more detail on those as well. And then finally, the main thing that leads to that actual pattern we saw in the last is those DNA fragments are then separated out by another process called gel electrophoresis, and then you can look at those actual patterns. So now to go through some of these kind of sub-processes that are involved in the DNA fingerprint, the first is polymerase chain reaction, or PCR. So it's called polymerase chain reaction because this is essentially DNA replication in a tube. So the polymerase that's being referred to here is DNA polymerase. So if you recall, DNA replication occurs in a strand of DNA where helicase will come in and open it up, forming what we see here, which is the replication fork, and then DNA polymerase molecules will come in and make a complementary strand, which at the end gives us two DNA molecules. So in PCR, or again polymerase chain reaction, what is going to happen is you'll have a small initial sample of DNA like we see over here, and then it's going to go through multiple cycles or rounds of replication where each time they're ending up with two. So you can see very quickly here, each of these molecules is leading to two more, that you have a huge amount of DNA at the end. People say this is amplifying the DNA. So PCR is again used to take a small sample and give you more so that you can look and see, what is actually in that sample. So once you have a lot of DNA, you need to cut that DNA up into smaller fragments so you can actually visualize it. DNA is too big by itself to do anything with, so they're going to use, the scientists are going to use restriction enzymes. And what these are, this is kind of interesting, they're actually different enzymes, and there are hundreds of them, that have been extracted or taken from bacteria. And these enzymes have specific sites that they recognize within the DNA. So here's an example down here where whenever they encounter that sequence in the DNA, they will actually cut the DNA. And what this ends up doing is it cuts that long chromosome, those long fragments of DNA, into fragments that are about 100 to 300 nucleotide fragments. And if you think about it, a chromosome is going to have thousands to tens of thousands of nucleotides. So this is going to be, basically chopping up the DNA into small sequences. And again, basically the restriction enzyme, it's going to cut it whenever it gets to that site. And for each of those different enzymes, it's going to have a different, what's called recognition site. So where it's looking and cutting that. So again, just to visualize this in a different way, I do want you to know that this is actually happening in a test tube. So when they do restriction enzymes, it'll either be in a large test tube or a little tiny tube. And again, you can almost visualize the restriction enzyme like scissors. It'll come in, and whenever it sees that site, it will cut, and the DNA will be in small fragments. Then you can, take it and run it out into gel electrophoresis, which we'll look at next, and then we can find our gene. So in gel electrophoresis, what's going to happen is that cut up DNA that was cut up with those restriction enzymes is placed in something called an agarose gel. And this looks like Jell-O pretty much. It's made of a substance that's very similar to Jell-O. And there's an agarose gel down here at the bottom right here. So that's what it looks like before you do anything to it. You cover this with a salt solution because you are going to then run an electric current through it, and salt helps to conduct electricity. The next thing that you do is load your DNA in. And so right here, all these little, what look like little rectangles, those are what are called wells, and the DNA is loaded into those little wells. DNA itself is negatively charged because if you recall, the DNA is made up of nucleotides, and if you remember, on the nucleotides you had the sugar, which in DNA is deoxyribose, and then attached to it you had a phosphate group. Well, that phosphate group is going to have a negative charge on it. And then you also have your DNA bases. So that would be something like cytosine that I just drew. So again, that's your base, that's your sugar, and then the phosphate is negatively charged. So when you load the DNA into these little wells, you put your little DNA sample in there. You're going to set this up so that you're going to run an electric current through here with the positive on this side and the negative on this side. What that will do is it will move the DNA from these wells towards the positive charge because, again, DNA is negatively charged. The other thing that this is going to be moved on is not only based on charge, because, again, DNA is negatively charged, but the size. So when you run this out with the electric current, those base fragments that are the smallest are going to run the fastest. So you'll end up with bands, when you look at it, where you have the smaller bands at the bottom and the larger fragments at the top. The other thing that people do when they do gel electrophoresis, kind of as we saw in that first sample, but it's been a little while, they'll run what's called a DNA marker with it that will help them look and compare the known sizes so they can see exactly what size their samples are. So after they've run this out, you take that agarose gel and you'll stain it with a specific chemical that when you then expose it to UV light, and that doesn't mean you take it out in the sun, you put it in a little chamber, and then they take that picture of it like we saw. That picture is actually the DNA fingerprint. So an example of how this DNA fingerprint is used to help identify mutations is to actually look at sickle cell anemia. So in sickle cell anemia, it so turns out that the site at which there is a mutation, and if you recall, this was a point mutation, coincides with the restriction site of an enzyme. So one of those restriction enzymes. So in this is beta globulin, which is the gene that is affected in sickle cell anemia. If you recall that there is actually our normal sequence here, and then we have a problem down here where we had a point mutation. What this does is it changes the DNA in such a way that the restriction enzyme, which has this kind of funny name DDE1, but this particular restriction enzyme, which normally would recognize this site in the beta globulin gene, which is what, again, is going to be afflicted in sickle cell anemia, cannot be recognized anymore. What this does is it allows you to very quickly see, using gel electrophoresis, to see if that person has sickle cell anemia or not. Because in the affected person, they do not have that DNA cut. So what'll happen on the gel, you can see it here, right here is our person, right here is our person that has sickle cell. They'll end up with a band that's very large. Okay, it's very large because it was not cut, whereas somebody that has at least one normal gene will have bands that are smaller because the DNA was cut. All right, the other way that we can visualize and see if someone has a mutation is to look at a karyotype. And this is essentially just a picture of someone's chromosomes. And those chromosomes just get matched up based on size and the little banding patterns here. And then you can also tell if a person has a chromosomal mutation using this as well as their gender. So part of this is cut off, but down here, this is a woman because that would be the X and the Y would be here, so this would be a man. All right, so first looking, just as a reminder, a deletion, that's where we had a piece of a chromosome that breaks off and it gets lost. So looking at a karyotype, and these are kind of difficult to see in some cases, here's an example of a chromosomal deletion of, this is on chromosome 18, and these are in people, again. So you can just barely see it, but you can see here that we're missing a little piece of this chromosome. So people that do this for a living, they're very skilled in it, but we are going to become skilled as well. So again, to see the deletion, you see just a little part of it is missing. Our other type here, our next type here, rather, is a duplication, and there the gene is copied and stays in the chromosome. So what this looks like on the karyotype, you can see it right here with the X. In this case, the duplication, it looks like some is added onto the end because this particular gene right at the end was duplicated. So it does lead to a lengthening, and a lot of times the duplications are visualized as well with some color, which we'll see in some of these other ones. The next type here is translocation, and as a reminder, that is when one broken piece on a chromosome breaks off and it swaps with another broken piece on a chromosome that's not similar. So you're going to have two totally different genes getting swapped. This is not a great thing. A lot of times these translocations can lead to infertility, cancer, as well as mental problems. So translocations, they have to be visualized using fluorescent markers so that you can really see them. And you can see here there was a translocation between chromosome 9 and chromosome 22 as visualized by the color. So that one's kind of easier to see. But again, it's a swapping, and you can see it in the karyotype. The next type here, an inversion. As a reminder, what happens in an inversion is you have your normal chromosome, and a piece of that chromosome, a lot of times it's in the middle, but it could be on the end or somewhere else, will come out, and then it goes back in, but backwards. This means the genes aren't in order anymore, and when that gene is transcribed and later translated, the information is incorrect. In inversion, this one's very subtle, but we can see it. If you look down here at this chromosome right here, this is where the inversion has occurred. So if you look at the banding here, this is actually flipped around, okay, because it should match this one. So when you look at all of these homologous chromosomes, you can see they have similar banding patterns. So that banding is going to help us see if something is wrong. So again, here, this whole portion has actually flipped, and that's led to the banding not matching. So that would be what an inversion actually looks like on a chromosome. And then our final one is a non-disjunction. So here, these are fairly easy to visualize because, as you recall, all a non-disjunction means is that the chromosomes did not separate correctly during meiosis. And this can either happen in meiosis 1 or meiosis 2. The result always, though, is going to lead to at least one of the gametes, so one of the daughter cells, having a trisomy, which is where you have an extra copy of a chromosome, or a monosomy, which is you're missing one copy. So the example down here is trisomy 21, trisomy X. But trisomy 21 is Down syndrome, which is the most common that we would see. So this is very easy to visualize. You just count. Here, this is a trisomy X because there are actually 3 copies of the X. And I think the X is being cut off for you guys. But what this says is 19, 20, 21, 22, and X. Okay, X, our sex chromosome, is the last chromosome. So there it's very straightforward. You just have an extra one or you're missing one. One thing I do want to point out to you, though, you do have to pay attention if you have a male. He's not monosomy. If he only has one X, that is something that a male should have. So with that, we are at the end of the lesson. There is a little extra thing that you need to do with gel electrophoresis that's linked in your notes. Make sure you do that and complete the full summary.